could go back to a time when the Earth was young, we would find a world choked in poisonous gases that spewed from a molten interior. Life could not survive on this scorched land. But in the oceans, things were different. 3.5 billion years ago, something began to appear that would change the planet forever. Surrounded by the protection of the sea, the first cells came to life. Today, descendants of this early life still flourish. We call them bacteria, and they've evolved to live in every habitat on Earth. With a deep-diving submarine, we can see one of the most unusual of these habitats, volcanic vents that split the sea floor. Here, the temperature reaches more than 300 degrees Celsius. Water pressure is nearly 2 tons per square inch. The prize for adapting to this hostile place is a rich supply of high-energy hydrogen sulfide gas that bubbles up from the vent. The bacterium processes the gas for energy, while bright yellow granules of sulfur waste collect in the cells. The carbon they use to build cell structures comes from the CO2 in the seawater. Of course, bacteria have adapted to less extreme environments as well. On land, they reside in and on almost everything. In soil, on plants, anywhere there's a food source. Using a plant as a source of food, however, does not necessarily mean destroying the plant's living tissues. Some bacteria have established relationships where both microbe and plant benefit, as in this case. These are bean root nodules. In each of these tough masses, millions of nitrogen-fixing bacteria live among the bean cells. For its part, the bean not only provides a residence, it also supplies the carbohydrate the bacteria uses as its food. In return, the bacteria performs a service which is achieved by few other organisms on Earth. It converts nitrogen gas from the atmosphere into the nitrogen nutrients required by nearly all living things, including the obliging bean plant. Bacteria have also found homes in the tissues and organs of animals. We know that bacteria are often permanent residents in the guts of many different species. But some animals, like these aphids, have developed special cells just to house their bacterial tenants. Deep in the abdomen of this aphid are a cluster of cells called the mycetocytes. With careful dissection, the clusters can be lifted out and prepared for a microscopic examination. Every individual aphid has many mycetocytes infected with bacteria. It seems that each aphid infects its own offspring while they are developing embryos. The light microscope reveals that the cells of the mycetocytes are jammed with thousands of spherical bacteria. Exactly what the bacteria does for the aphid is not clear, but it must be something vital, because if the bacteria are removed with antibiotics, the aphid's offspring are undersized and sterile. Dehydrated, sprayed with gold, and mounted on a small stub, the aphid cells are prepared for examination with an electron microscope, able to magnify many times greater than the light microscope. The electron microscope can show new details about shape and arrangement of the bacteria cells. This bacterium is unique to the aphids. Their partnership seems indivisible, probably because the evolution of each animal has been intertwined one with another for thousands of generations. The modern scientific study of bacteria relies on a few basic research techniques. First, all glassware and tools must be sterilized to ensure that experiments will not be contaminated with unwanted cells. At each step, this sterilization is repeated to guard against an accidental brush with an unclean surface. After heating, a thin metal wire called a loop is inoculated with a few bacteria. The opening to the test tube is passed through the flame to destroy any bacteria that might be threatening escape. The bacteria-laden loop is then streaked on a dish filled with a gelatinous substance called agar. Mixed with the agar is food for the bacteria. This agar-nutrient combination is called the culture medium and is one of the most important tools in bacteriological research. Culture media allow scientists to grow and isolate different types of bacteria at will. By simply varying the nutrients included in the agar, specific species of bacteria can be selectively grown. Once a dish has been streaked, it's incubated at a controlled temperature, and the bacteria multiply. After hours or days, thousands of bacterial colonies appear on the dish. The tracing of the original strokes of the loop can still be seen in the pattern of colonies. The growth of bacteria can be staggering. In hours, a sample can go from a damp smear of liquid to a lumpy surface thick with millions of cells. Each individual colony was started by a single cell that doubled and redoubled itself to become a visible mass. To get a look at a single bacterial cell, you'll need a good light microscope, one capable of 1,000 times magnification. When they come into focus, you'll see that bacteria come in 4 basic shapes. There are cells called spirillum that are shaped like a coiled rod or a corkscrew. There are also roughly spherical cells called cocci. Cocci can be arranged in clusters or chains. The third shape is the bacilli, rod-shaped cells that come in pairs, chains, and other arrangements. Finally, there are long, thin, thread-like cells called filamentous bacteria. Scientists use shape, size, movement, and nutrition to characterize bacteria. Modern classification places bacteria and other prokaryote cells in a separate kingdom, the kingdom Monera. Using an electron microscope, we can see some of the features that make prokaryotes different from the other major cell type, eukaryotes. For one thing, prokaryotes lack the true nucleus, as well as other membrane-bound organelles like chloroplasts and mitochondria. Instead, they have a continuous loop of nucleic acid that lies in direct contact with the cytoplasm. This chromosome carries all the genes needed for normal growth and reproduction. In addition, a portion of the DNA in bacteria can also exist as separate, smaller segments called plasmids. Recently, researchers in genetic engineering have found a special use for plasmids. It turns out that the genes from another creature can be spliced into the DNA of a plasmid. In this way, a bacterium can be modified to manufacture drugs and other important chemicals in great quantities. Some bacteria have cell fibrils called pili, which they use to attach themselves to surfaces, such as other cells. Motile bacteria have another type of cell fibril called flagella. A cell can be adorned with one or dozens of long, snaking flagella. By rotating the flagella like a propeller, the bacteria push themselves through the medium at a very respectable rate. A few bacteria have developed a less vigorous alternative to flagella. They glide along on a slippery layer of mucus, much in the same manner of a snail. It's a graceful, if not rapid, mode of travel. The variety of bacterial lifestyles is staggering, from animal-like flesh eaters to plant-like photosynthesizers. Bacterial photosynthesis, however, can have several unusual twists. Take purple sulfur bacteria, for instance. These cells have adapted to endure the scalding waters of hot springs, reinforcing their cell walls against the fierce heat are special protein. Purple sulfur bacteria are photosynthetic, but their biochemistry is very different from that of green plants. Rather than water, these bacteria use hydrogen sulfide as the electron donor in the photosynthetic cycle. The result is, rather than liberating oxygen gas as a byproduct, these bacteria produce tiny grains of solid sulfur, these dense yellow particles. It has been said that if it's edible, there's a bacterium that will eat it. And that's not far from the truth. There is at least one bacterium specialized to metabolize just about every type of organic material. The broad appetite of bacteria is the basis for their role as the primary decomposers on Earth. A bacterium important in the decay of wood and leaves is Myxobacteria. Colonies of these cells glide along the forest floor, absorbing nutrients from the fallen timber. When food becomes scarce, the cells pile one on another, forming a tree-like structure called the fruiting body. Spores at the top of the fruiting body are carried by the wind to colonize greener pastures. Organic decomposition is not a process that happens only under the leaf litter of a forest. We rely on decomposers to recycle much of the waste we create. A landfill, like this one, is really a centralized, organized site for decomposition. Food scraps, garden clippings, even newspaper can be broken down by bacteria. Here, bacteria with the enzymes to metabolize cellulose attack the wood fibers in a sheet of newspaper. To be effective, the cells must attach to the fibers and release their digestive enzymes onto the wood. In many ways, a landfill is a self-contained ecosystem with its own food chain. For example, chemicals produced by the cellulose-eating bacteria are fed on by methane-producing bacteria deeper in the fill. Another place methane-producing bacteria thrive is here in the rumen or stomach of cattle. This steer is equipped with a rubber trap, a kind of window into the animal's stomach. It's a painless way to allow researchers to study stomach contents. The steer's stomach is full of what you would expect, grasses and hay. But it turns out that the steer could not digest the grass if it weren't for a community of bacteria and protozoa that live in its rumen. As in the landfill, one of the bacteria living in this murky world is a methane producer. In a flask like this, methane evolving from the steer's stomach juices collects and fills the container. Methane is a light, combustible gas that's a waste product of the microbe's metabolism. Striking a match confirms that the bacteria are hard at work. Under the microscope, the menagerie of residents of the steer's rumen can be examined. Protozoa stir up the grassy soup, keeping the bacteria mixed and feeding. What happens naturally inside of a steer, humans have been able to recreate and put to work in processing sewage. We produce billions of gallons of liquid waste. But what's waste to us is food to many types of bacteria. At sewage plants like this one, active cultures of microbes are added to the slurry of waste. As in the cow's rumen, the cells form an interdependent community. Mats of filamentous bacteria absorb the soluble nutrients, while the activity of protozoa keeps things well mixed. Part of the reason bacteria have been successful in so many different environments is because of their tremendous reproductive output. Bacteria reproduce by binary fission, and a single colonizing cell can soon give rise to thousands of descendants. Just how fast do colonies of bacteria grow? Some have doubling times of 20 minutes. That is, every 20 minutes, the number of cells in the culture doubles. Rapid reproduction makes bacteria well suited for their main ecological role as decomposers, but it also makes them a danger to unprotected food supplies. Preventing food from spoiling really means preventing bacteria from growing. The most fail-safe method is to simply destroy all the bacteria in and on the food. This is the idea behind heat pasteurization and canning. Food is first cleaned and sealed in airtight cans. The cans prevent any bacteria from reaching the food from the outside. But what about the bacteria trapped inside the cans? They're destroyed when the cans are heated to high temperature. The importance of killing all of the bacteria in the cans cannot be emphasized enough, because the airtight world created in the can, perfect environment for many microbes, including this one, Clostridium botulinum, the agent of botulism. Clostridium produces a potent toxin that can cause fatal paralysis. What makes Clostridium especially dangerous is its ability to retreat to a hardy spore form. If heat pasteurization is not complete, dormant spores can come alive, and the bacteria starts to grow. The illnesses associated with bacteria do not stop with food spoilage, of course. Disease bacteria, or pathogens, are responsible for a long list of ailments, including typhoid, cholera, and pneumonia. These, in fact, are pneumonia cells, and massed in a protective gelatinous capsule. The capsule is erected by the bacteria to ward off attack by cells in our immune system. Here, rod-shaped bacteria, colored green, are being pursued by immune system phagocytes, colored red. When the phagocyte overtakes the bacteria, it engulfs and dissolves the microbe. The phagocytes in our immune system tend to be very busy because bacteria are persistent and ubiquitous in our world, part of every meal, every breath, and part of every touch.